Welcome to the first presentation on communication, integration, and homeostasis. This is the first of two presentations on this topic on human physiology. And we begin with a brief overview of the chapter. We'll talk about cell-to-cell communication, signal pathways, novel signal molecules, modulation of signal pathways, and control pathways, looking at response loops and feedback loops in these presentations. We begin then thinking of an overview of the cell-to-cell communication. This is very important in multicellular organisms, such as the humans. You'd be impressed by the total number of cells estimated to be in a human body, as noted in the textbook. So this type of communication is extremely important. It can be a cluster of cells communicating to thousands or millions of cells. So it's really quite impressive when you think of the numbers, and obviously very important to survival. So the physiological signals that we might consider then would be electrical signals. So these would be changes in the membrane potential of a cell. You should review membrane potential, resting membrane potential, and physiological mechanisms for creating it, generating it, and maintaining it in the earlier chapters of the textbook. The chemical signals would be the other, secreted by cells into the extracellular fluid, also called the interstitial fluid, between the tissue cells. And of course, that fluid is within the space of the same name, the extracellular space or the intercellular space between and among the cells. So the chemical signals will be responsible for most communication within the body, as we know. The target cells, this is a key term that we want to put in our mind. The signals don't have much effect unless there's a target, and the targets can be receptive to those signals. There are four basic methods of communication that are listed in the textbook, and we'll go through them each separately. So this is an example where in the textbook they will list, for example, these as a set of four, and then begin discussing them. Here in the presentations, we don't list them and we go right into them. So the assumption would be that a typical type of exam question or assessment question would be whether you can recognize and recall those four from a list. So the first cell-to-cell communication under methods is the direct contact and local cell-to-cell communication. So cells that are immediately adjacent to each other, whether they are destined to be such, like the cells in our skin, or become approximated due to movement of the cells, like blood cells bumping into each other or into the cells lining the blood vessels as they're flowing. The cells are like lymphocytes. Immune cells are flowing through the body tissues. So, for example, in the heart tissue, we'd see many gap junctions for the type of discussion that we're considering now. We'd be less likely to see them between a white blood cell and a cell lining a blood vessel called an endothelial cell. So the gap junctions would be the first ones. These are probably going to be associated between and among cells that are fixed in place, like the cardiac muscle cells. So there'd be transfer of both chemical and electrical signals, so the functioning of the heart so it can contract the way that it does depends upon appropriate functioning of the gap junctions and having the correct number of gap junctions. These form direct cytoplasmic connections between the adjacent cells for signaling. Continuing under the cell-to-cell communication methods, we see the cell adhesion molecules, or CAMs is the acronym for that, and these are the contact-dependent signals. So this is the case where I was saying maybe the white blood cell in the blood bumping into the endothelial cell. If the endothelial cell, we would consider that to be the one on the right-hand side, is expressing a protein on its surface that will cause a stickiness or will bind to a specific protein on the white blood cell, then the white blood cell will be likely to stick and slow down in that area of the blood vessel where the endothelial cells have that receptor. and they're most likely to stick there. Once they bind, then they might pass through into the underlying tissue. Say it's a pus cell, white blood cell that goes into an area where there's an infection to produce pus. So these cell adhesion molecules can actually transfer signals in both directions. So not only is it just for the cell to stick, but then you have to signal to allow for the next stage of the event. The stickiness is there for a reason, whether it's good, to help fight off an infection, or part of a pathology such as sticky blood vessels and sticky white blood cells in diabetes or in people with high cholesterol and everything, and develop hardening of the arteries, the atherosclerosis. So obviously the integrated approach in this course brings these basic ideas back into play as we get into specific areas of discussion. So I won't give so many examples anymore as we proceed through these slides. So the contact-dependent signals require interaction between the membrane molecules on two cells. The direct contact and local cell-to-cell communication is a variant on this. It is the third, the autocrine signals and the paracrine signals, where the cells are in an area where other cells are nearby. The source cell, so there's a source that synthesizes and secretes, and then there's a target, either itself for autocrine or a nearby cell for paracrine, so parallel to it or in the relatively immediate vicinity. Depends on how far this this chemical signal, this extracellular or intercellular, between cells, chemical messenger can diffuse. Remember, it may be a molecule that is degraded very rapidly by other molecules like enzymes in the extracellular matrix out here, and so it may be capable of diffusing if it doesn't degrade long distances, but it may not be able to get through physical barriers, thick connective tissue, or one loaded with enzymes that are going to break it down. So it depends upon the nature of the tissue and what the response is that is required for the chemical signal. The autocrine signals act on the same cell that secreted them. Paracrine signals are secreted by one cell and diffused to adjacent cells. So if you think of getting bitten by a mosquito and you get a little red bump that's localized, this is what you want to think about. It's a local event. It's not quite such a systemic effect unless you're deadly allergic to bees. You're only going to get a very local effect, a little red spot in the skin. But if you're very sensitive, then somehow the signal amplifies and spreads throughout the entire body and you get anaphylaxis. So again, I can't resist applying them here and there, but I don't want to get into it to any great extent, just to keep you thinking about where these might fit in later discussions. So paracrine and autocrine are chemical signals, and we might call these, use the plural, paracrines and autocrines are chemical signals, and hormones are secreted by endocrine glands or cells into the blood. So they only target cells with receptors for the hormone, the only cells that are considered target cells are those that express the receptor for the hormone on the surface, and it's through that receptor, obviously, that the target cells can respond to the signal. So here we see the hormones typically used for chemical messengers, extracellular, intercellular chemical messengers that are going to be carried great distances. So there's some diffusion, and this is where I like to make the point that the hormones are released into the interstitial fluid, and then they diffuse into the plasma, the blood tissue, aqueous portion, right, the plasma, and then are washed away and diffuse out at other locations into the interstitial fluid and encounter all cells there, and only can affect the target cells, meaning that the ones that are expressing the receptor protein or the receptor either on the surface or inside the cell will talk about these differing mechanisms of being receptive later. And those would be the responsive cells. But all other cells are bathed by it. So this is the point to remind us of a local concentration generated here and a concentration gradient that is maintained across the blood vessel wall by the blood moving through that area. So this high concentration gets washed away and is replaced by blood with a lower concentration relative to the interstitial fluid nearby. So this is the mechanism. This diffusion gradient, chemical gradient, is the mechanism for removing, if you will, the chemical messenger from the interstitial space, where it still may be having autocrine or paracrine effects, depends upon the hormone and the overall function. That effective local concentration is going to be reduced by what is washed away with the blood. And then at other locations where you don't have the hormone being produced, the interstitial concentration is going to be lower, so you now have the opposite concentration gradient higher in the plasma than it is in the interstitial fluid, so it'll diffuse out. So these diffusion gradients, whether they are chemical or electrochemical, are extremely important for the movement of chemicals throughout the body. So that's a good point to make that, and we see it again here in this type of short distance chemical messenger diffusion, similar to the paracrine and autocrine, but this is a different type. It's the synaptic type, where a neuron has a termination close to the target cell. But we are actually looking at this, so this is diffusion dependent, but we are actually considering this signaling along a neuron as a great distance as far as talking about cell-to-cell communication. So this cell somehow makes a determination back here near its nucleus that a signal needs to be sent to the target cell. And so it sends the electrical signal. Remember, we talked about the membrane properties for generating electrical signal that is coupled to the release of a molecule that then is in the interstitial space and can diffuse toward a target cell. So again, the concentration gradient is going to be highest at the point of release at the axon terminal, and should be lowest over here near the target cell. And there are means of removal of this transmitter from this space, either through degradation, as we said, or diffusion away and wash out by the blood if capillaries are nearby. So you always have the input, the activity, and the removal in each of these cases where you have a target cell response that is dependent upon the concentration of the molecule in the nearby interstitial fluid. What we want to do is remind ourselves at this point, for a very important concept, it's an essential concept of appreciating basic presentations and illustrations versus the real-life event. So you have a certain concentration of a molecule nearby, a binding site, the receptor, and there is a statistical probability based upon the concentration of the molecule that can bind to it. It's called a ligand, L-I-G-A-N-D, and the binding site, the receptor. So you have a concentration of both of them. And so if you have a very high concentration of these red molecules, the chemical messenger, and a medium concentration of receptors, you're very likely to have all the receptors occupied at nearly any one point in time, and this target cell is being bombarded. If you have a million receptors here and only 4 chemicals, it's very unlikely that at any one point in time, any one of these 4 chemical messengers is going to be randomly bumping into a receptor, even though they're all over the target cell. So the target cell is ready to respond. It might be hyper-responsive, but there's not enough chemical messenger to do it. So we'll get into that regulatory system later, but again, as we encounter these, I like to bring it to your attention so you don't get too distracted by these simplified illustrations. But they're critical to help us understand the basic things of generating a chemical signal that is coupled to an electrical signal. So one final point before we leave this. We also want to think of this electrical signal as a digital signal. And the chemical messenger is an analog signal. It can be any concentration from 0 having any value up to the highest value possible, right? Whereas, as you might know, the action potential coming down the axon, this electrical signal, is an all or none. So it can't be, greatly amplified to any degree or decreased to any degree. It's an all or none, so it is a digital square wave type of an event. And then that's converted to the analog event with the release of the chemical here. So this analog broad range of concentration of the extracellular chemical messengers is important to have the kinetic properties that we talked about related to the concentration of receptors nearby on a single target cell or a population of target cells to generate some probability that the target cell or cells would be responding to any degree based upon receptor occupancy. So I hope you followed that okay at this point, and we'll be re-emphasizing it as we go through. And so now let's just push on through the remaining slides. So neurohormones are a bit different. They are the same in the local area of release near a capillary, that you get the interstitial concentration greater than the plasma, so diffusion gradient into the plasma. You also have the coupling of the digital signal, the action potential, the electrical signal, with the release and a conversion of digital to analog. So you've got a D to A converter, digital to analog converter system here. And the analog signal ends up as being some range, a broad range of concentration possibilities of this neurohormone in the plasma, where it's distributed throughout the body, and you have thereby a plasma concentration greater than the interstitial fluid. And again, it's the same thing. At the remote location from the site of synthesis and release, the source site, at that remote location you generate an interstitial fluid concentration where if you have some receptors, it can have an effect on the target cells. If you have 0 receptors and 10 million, 1,000, billion, whatever chemical messenger molecules nearby, the really high concentration, you can't have an effect on the cell because it doesn't have the receptor. So this is an example of going from 0 to 1 receptor where you have no response and some response. Anyway, it helps us to make that point. So neurohormones are chemicals released by neurons into the blood for action at distant targets. But it's still the coupling of an electrical signal that can go over a long distance to the site of release. So you've got the electrical signal coupled to the release. Continuing on with these methods, the cytokines are an example that can act as both local and long-distance signals. Interesting discussion on this topic in the textbook. All nucleated cells synthesize and secrete cytokines in response to stimuli. So this makes them a bit different because we don't have a limited, highly specialized subset of source cells. Every cell is a source cell. And in development and differentiation, cytokines usually function as autocrine or paracrine signals, so then nearly every cell is a target cell. So this is very important, and the cytokines include the chemical messengers that can cause the global systemic response to a bee sting if you are hypersensitive to it, to bring about a whole body response of anaphylaxis, right? So every cell in the body can react, and that's what makes them so universally important for whole body signaling, but also potentially dangerous and lethal if it's not working well. So if the homeostasis is offset, if there's some type of a pathology or disease or disorder, then there's a danger to the organism, to individual cells, and then collectively to the organism. So in stress and inflammation, some cytokines may act on relatively distant targets. So the inflammation is the type of response that's out of whack when you have the hypersensitivity and possibly anaphylactic response to a bee sting. So now we are beginning to think about signal pathways, and we have an overview. And here we are thinking in this overview of the source. We have the signal molecule, and then ultimately the response. So the signal molecule binds to the receptor protein and activates an intracellular signal molecule set, or intracellular signal molecules, and these subsequently will alter the target proteins within the cell and create a response. So we are now thinking about the extracellular signaling, the extracellular signal encountering the target receptor, and we get a signal transduction from outside of the cell to the inside of the cell, and now we generate a whole new set of signaling molecules, intracellular signal molecules. And then they have targets within the same cell to bring about the response. So chemicals talking to chemicals to bring about changes based upon the information from the original source is one way to think about this. So let's think about the receptor locations. These are the very important molecules. If they didn't exist, there'd be no coordination of the chemical events in an organism. So the target cell receptors can either be on the membrane, as we had talked about. I used the term expressed these receptor proteins on the surface of the cell. That way, a lipophobic or hydrophilic signal that cannot just slip through the membrane, as can the lipophilic signal molecules, will be restricted to binding sites that are expressed on the surface of the cell. Some lipophilic signals, because they're still out here in this same fluid compartment, typically interstitial, but it could be in the blood too. The blood plasma is another subset of extracellular fluid that is separated from the interstitial fluid. So extracellular fluid includes both of those two, the interstitial and the plasma. So this could be plasma as well, where a cell in the blood is being signaled by either a lipophobic or a lipophilic signal molecule. The lipophilic signal molecules have the other option to freely diffuse through the lipid bilayer membrane. So if they can dissolve in lipids, they just get right through. And the receptors, proteins for these types of chemical signals are located either in the cytosol or in the nucleus. Those that are in the cytosol, as we see, once they are ligated or bound to the signal, then they, almost every case, will be given permission to move into the nucleus and then act on the DNA. So one way of thinking, a classic way of thinking about the lipophilic molecules that depend upon changing the expression of genes to generate a response, is that that's a slower response related to changes in the gene activity, the rate of gene expression to produce messenger RNA and then the proteins. Then the proteins have to be activated, processed to be activated to produce the response. It's also at this point that I like to remind people that these types of receptors on the surface, we will see, can bring about very quick, immediate responses in a cell. But whenever a cell responds, it needs a signal to get to the nucleus to say, you just responded to something and you burned up some proteins or other molecules that depend upon proteins to generate them. So you need to make more of that protein, so you get gene expression changing there as well. Or other ways that this type of a receptor mechanism can have a slower response. So let's rethink this then. The lipophilic signaling typically is a slower response unless it's on an immediately active receptor on the surface of the cell. The lipophobic signaling through a membrane receptor mechanism typically will have a fast response, and coupled with it some type of a delayed response, whether it's just one that needs to replace what was just expended to generate the response, or to coordinate other phases of the response that would be delayed. Right. So a cell doesn't always have to have just an immediate response to something. Sometimes we want an immediate response with a follow-up, right? So if you are just about in an auto accident, then you want to respond immediately to prevent it, and then also you need to have the follow-up to correct where you are to get back in your lane and back on traffic, online in the traffic, to keep things going where they're supposed to, back to homeostasis. So there's a lot of coupling of those types of events. The lipophobic signal molecule in the extracellular fluid can be interstitial fluid or plasma, as we said, depending upon where the cell is. And there is the cell membrane receptor that becomes ligated. So the ligand receptor complex. So a ligature is where you pull two things together to stick them together. So a ligand is something that sticks to something else. So it's the binding molecule that binds to the binding receptor, the binding site. So in the ligated form, you can get a rapid cellular response transduced to the intracellular fluid. So we have this transmembrane signal transduction from the extracellular to the intracellular compartment. So these can be rapid, and the rapid can be coupled with a slower phase of the event too. So in this idea of the signal pathways, we think of the membrane receptors first, and there are 4 categories of membrane receptors. We see them summarized on this slide. Receptor channel. So we might think, gee, this is only going to have an immediate effect or response from the cell, but whatever comes through the channel might end up having some intracellular effects that can be coupled to a more delayed response. So let's not always just think of these as having only an immediate effect. We want to ask, can there be a delayed effect as a follow-up to what just happened in the short term, the response. And then there's the receptor enzyme, so a protein expressed on the surface of the cell for an extracellular chemical messenger, most commonly for the water-soluble, the lipophobic or hydrophilic, the peptide or protein types of signal mechanisms. And the lectures in the endocrinology will talk about that as well. These can be coupled directly to or have as part of their total protein amino acid sequence sites that end up on the inside of the cell that have an enzyme property. So when they are ligated, you get a change in the enzyme activity inside the cell right near the membrane, on the underside of the membrane. Another set of signal molecules can bind to receptors that are one protein or a series of them, the subunits, that snake in and out of the membrane, having transmembrane segments. There's probably 7 here. 1, 2, 3, 4, 5, 6, 7. There's a whole family of membrane protein receptors that have 7 transmembrane spanning segments. So obviously there's a great more detail than what we're getting into here, but I just want to draw your attention to that at this time. So you've got all these different portions, and they're all named and everything. And there might be 2 or 3 proteins like this together, so don't be misled by the simplicity of this. But they alone often don't have any capability of responding other than undergoing a conformational change when they are ligated, and then that conformational change will change the affinity for these G proteins and excuse me, and the binding, the ligated or unligated state here, where the conformation of these will change subsequently. So this is a very interesting protein cascade of just a bind there, a twist in the protein or proteins, and then a change in these proteins, and then a change in activity. So this is the G protein coupled receptor. And then finally, an integrin receptor. Here we see again it's transmembrane spanning, but there's only 2 transmembrane spanning portions in this receptor complex through the cell membrane. So an extracellular portion to be ligated, and an intracellular portion for the transmembrane signal transduction. And the integrins are typically associated with some type of anchor protein complex linked to the cytoskeleton inside the cell for motility, changes in shape, or securing the site of binding to the extracellular matrix so that the cell will not move, right? So, or it might be the reverse, to loosen up the grip with the extracellular matrix so that a cell now it can become motile. So this obviously is something very important in cancer, where you can have a metastatic type of a cancer where the cells don't stick in one location, they just freely move around, and then they decide to stick anywhere else in the body. So it's a very important concept to understand, and has a high clinical relevance. So if we can control it some way, we might be able to control selectively just the spread of cancer cells. That would be a tremendous discovery. So the signal transduction will convert one form of signal into a different form. So we already talked about digital to analog. You may be aware that just recently the television signals changed in 2009. You see that this is a copyright of 2009, so that's the year of recording. There will no longer be analog signals for television. Everything had to be switched by federal regulation requirements to digital. But you might have an analog television, so you've got to get a D to A converter to hook to your analog television now to convert the digital television waves to analog signal for your television if you don't have a digital television. So that is a recent event that helps us to understand not only just the basic idea of transduction, but also the analog to the digital. So the digital is restricted incremental changes, a square wave type of a change. And the analog is a free range, any value in between. So you might have a digital signal that can only ramp up in 0.1 increments. An analog signal could ramp up in any increment of signal, 0.00000001 or 0.09, whatever it would be. So that's a distinction. So again, we are thinking of an external signal through a receptor coupled somehow either within itself or with other proteins to a transducer, and then some type of amplifier that can bring about their response. So some systems may have an amplifier or may not have an amplifier, but that is a very important aspect in most cases. So let's think about this idea of the signal amplification then. where it is really important. So the transducers convert extracellular signals into intracellular messages with which create a response. So the receptor complex activates an amplifier enzyme, and we're going to follow through this series of events in the next set of slides to look at the signal transduction, the transmembrane signal transduction from extracellular or intercellular signal source, the ligand, to the receptor, across into the intracellular compartment. Well, I guess it's not in this set of slides, but ultimately we do look at that in the series. So the signal amplification is brought about by the amplifier enzymes. So if you activate one enzyme, it can take a lot of substrates and activate them as products that then can become signals within the cell. So let's consider the three most common amplifier enzyme signaling mechanisms: adenylyl cyclase, guanylyl cyclase, and phospholipase C. And think about how we get from that enzyme to the second messenger. So the extracellular chemical messenger is the first messenger, and then there's going to be some second messenger inside the cell. There might be third and fourth chemical messenger systems, but we're thinking now just about the secondary because it's going to follow immediately from the primary, or the first chemical messenger, the extracellular one. So here are three examples, actually four, two for one systems, but examples of the three signaling amplifier systems that we talked about, or second messenger systems. There's the cyclic AMP, adenosine monophosphate, cyclic GMP, guanosine monophosphate, inositol triphosphate from a membrane phospholipid, and the diacylglycerol. So you know what a triacylglycerol is, or tri-fatty acid. It's a glycerol with three fatty acids. Clip one of them off, IP3, and you're left with diacylglycerol. Right? So they have those acronyms defined here below. So the amplifier enzyme, as we said, for example, adenylyl cyclase, I always want to say adenylate, is localized to the membrane. This would be the cytoplasmic membrane surrounding the cell, more toward the inside of the membrane as a peripheral protein. And the G protein coupled receptor is how it is activated, and it has as a substrate ATP and a product CAMP, cyclic AMP. Guanylyl cyclase is in the membrane, but also in the cytosol. So two locations. It is functioning at the membrane as a receptor enzyme, and in the cytosol for the production of nitric oxide, a chemical messenger, typically paracrine or autocrine, or both, and it converts GTP to cyclic GMP. And the phospholipase C is in the membrane, a peripheral protein, more on the inside, the cytoplasmic side, and it is a G protein coupled receptor that converts membrane phospholipids, as I stated, into inositol triphosphate and diacylglycerol. So let's consider the signal pathway, the biological signal transduction then. It converts chemical signals into cellular responses. So this obviously is an extremely important concept in physiology. So here we see two types of receptor mechanisms, the ion channel or ionotropic type of receptor, and one that is going to affect metabolism, the metabotropic type. The metabotropic in this case can have some modulatory effect on the ionotropic. So this is where we're beginning to build on the complexity of the interaction between these short-term and long-term effects. So this might be one way where you can have the coupling of a short-term ion event by this membrane receptor and the more delayed one based upon enzyme activity that has gene expression changes as part of the cell response. So just in general, let's consider this signal molecule. So we're going to convert the chemical signal into a biologically relevant response. So it binds to the membrane receptor, initiates the signal transduction by proteins. We can have amplifier enzymes or a direct activation of an intracellular signaling system that does amplify as well, but it bypasses this particular second messenger molecule that can also act on a similar population of cell proteins. These are protein kinases. There's many different types of protein kinases. Whenever you see kinase, you think of an enzyme that puts a phosphate onto a cell. There are phosphatases that will remove that phosphate later. Every biological event has to have something that'll reverse it or terminate it, right? So if a protein kinase puts phosphate onto a protein, it is a phosphorylated protein, then it may be activated and bring about the cell response. You need to terminate the cell response, so it has to be dephosphorylated by some mechanism, and then there's phosphatases that would be doing that. The amplifier enzymes producing second messenger molecules might act through protein kinases as well, a different subset or possibly the similar ones. And it may also be acting to increase intracellular calcium by binding, as we'll see, to the endoplasmic reticulum, protein on that receptor. Protein on the ER membrane exposed to the cytoplasmic side. And, excuse me for clearing my throat there, this increased intracellular or cytosolic free calcium that has to be regulated very tightly, because too much or persistent presence of a high calcium can kill a cell. So this is a very dynamic process of regulating the intracellular cytosolic free calcium. So there are calcium-binding proteins. Many of those are activated when the calcium binds to them, and it brings about a cell response. Excuse me again. So this might be coupled to an ion channel event as well, either modulating it, so it let's consider it in three ways. First, it may open or close it to have an immediate effect on the ion. Or this might depend upon a ligand as well, and it may modulate it, make it more reactive to the ligand. So a little bit of ligand with this one modulating it, and you get a bigger response from the cell. Or it might be the opposite, modulate it so that it doesn't matter how much you have of the chemical signal for this, considering it to be a ligand-dependent ion channel. And you just can't get a bigger response no matter how much you try to bombard it with higher and higher concentrations of its ligand. So there are many different aspects of that we need to think about rather than just the simplified model shown here. And I'm not trying to overdo the complexities. I'm trying to minimize the reductionism so you don't think of it in just one little way. We're trying to get these basic concepts that are essential so we can understand how they are expounded and modified and even complicated by cells, by biological mechanisms or systems. So signal transduction, there are steps in the signal transduction pathway that form a cascade. So we have the initial stimulus that can bring about the activation of an original system or molecule, like an enzyme, that will then activate a second in the cascade, that will then activate a third, that will then activate a fourth, so kind of a domino effect. And then this final one is a substrate that is acted upon to generate a product that now might be active, or it might be an essential component within the cell to bring about an appropriate response. It could be the breakdown of glycogen to release glucose by the liver, as an example. So the signal pathway, we are thinking about receptor enzymes in this case then. So tyrosine kinase is an example of an enzyme receptor. So here we have the extracellular fluid, the intracellular fluid. It's typically shown this way with the out up on the top and in on the bottom, and the cell membrane in between. So we have the transmembrane spanning protein, the integral protein. The extracellular exposed portion has the binding site for the ligand, and there is some type of mechanism for a conformational change when it is ligated that reverses when it is unligated, such that the enzyme activity, in this case a tyrosine kinase, so it will phosphorylate proteins at their tyrosine residues. So tyrosine is an amino acid, so wherever there's a tyrosine in the protein that can be phosphorylated, it has to be exposed at a certain way at the edge of the protein so that this enzyme can put a phosphate on there. So the amount of activity in this portion of the receptor molecule depends upon the frequency of being ligated. So here at this point, as we're transitioning to see the events related or associated with the ligated form of the receptor, let's remind ourselves that this is a dynamic binding and release that is going on. And it's the frequency at which, the average time over which you are more likely to have the ligated than the unligated form of this receptor that is correlated with the average activity of this enzyme, therefore a buildup of its product, an activated phosphorylated protein. Excuse me. So we don't just inject someone with adrenaline and then the adrenaline goes to the adrenaline receptor and just sticks there and stays there and sits there. It just adds to the concentration locally so that you have binding and release more frequently, because whenever this one comes off, there's going to be another molecule of the epinephrine or adrenaline to stick to it in the next instant of time. So over a period of time, you're likely to see it more frequently in the ligated form. So on the average, it is ligated more often than it is unligated, and therefore on average over that same period of time, you have more enzyme activity and you have greater production of the phosphorylated protein and a buildup of ADP. Right? So the signal molecule binds to the surface receptor, activates tyrosine kinase on the cytoplasmic side, and then you have a phosphorylated protein. Okay, but it is always the concentration of substrates and active enzyme that is related to the amount of product. Right? So if for some reason you don't have any ATP, I don't care how active this enzyme is, if you don't have the right concentration of substrates, you're not going to get product. But at any one point in time, you should be able to measure a certain amount of product and a certain amount of the substrates and a certain amount of enzyme activity. And they will all change one direction or the other depending upon the frequency of ligation of the molecule over time. So again, these static reductionist models kind of mislead us. So I don't want you to be misled from the beginning. I want you to take the time to rewind this and listen to that again if you need to, and to think about some of the chemistry and go back and look at the introductory chapters on enzyme activity and substrate and protein and enzyme concentrations and your biochemistry if you've taken it. Or think of it again hard when you do take your biochemistry course. So the G protein coupled receptors, or the acronym for that is GPCRs, are considered in this signal pathway slide. So we have the membrane spanning proteins. There's a cytoplasmic tail linked to the G protein. Remember, we saw the 7 transmembrane spanning segments in that other illustration. And there's a 3-part transducer molecule, the G protein complex. When the G proteins are activated, they open ion channels in the membrane, that's one possibility, or they alter alter enzyme activity on the cytoplasmic side of the membrane. So I think this is where we go through the series, and I'll read each of these as we do that. So let's consider the first GPCR, the adenylyl cyclase cyclic AMP second messenger system, and assess to some degree, introductory level, the amplification. So step 1, the sufficient concentration of ligands such that over a period of time we find the extracellular binding site more frequently in the ligated form than in the unligated form. In other words, enough molecule shows up to activate the receptor. So the signal molecule binds to the G protein linked receptor, which activates the G protein. There's actually some intramembrane movement. If you remember the fluid mosaic model, what happens is the conformational change and the affinity changes, and there might be some binding of other molecules. Changes in affinities, changes in bindings, and even some lateral movement, as they're indicating here. The protein turns on the adenylyl cyclase and amplifier enzyme. So it is inserted into the membrane, but it's really a peripheral protein. They're showing it exposed to the outside, but I'm not so sure it really spans the full membrane. Doesn't matter. Excuse me again. So number 3, plenty of substrate nearby, hopefully, ATP, and this enzyme that is now more likely to be in the active state will convert more ATP to cyclic AMP. So in the situation where there's a low concentration of the signal molecule, there's a low frequency of activation, so you're more likely to see this inactivated than activated. Same then with this molecule. So there'd be higher amount of ATP and lower amount of cyclic AMP. As you increase the concentration of the signal molecule, you more frequently see it in the ligated and activated form, where it is now dissociated from the G proteins because they're spending more time over here, and they keep this enzyme active. So you are going to see probably a local decrease in ATP, but definitely a local and intracellular increase in cyclic AMP. So the adenylyl cyclase converts the ATP to cyclic AMP, and then the cyclic AMP activates protein kinase A. The A is for the cyclic AMP here. There's going to be a protein kinase C, so we're pointing that out at this time. Link this pKa, acronym pKa, protein kinase A, to the A in the acronym for cyclic AMP. And then protein kinase A phosphorylates a range of different proteins, and many of those, as its enzyme activity is sustained by the higher frequency of ligated state, right, it cascades through. At each of these stages, there's an enzyme that inactivates this molecule. There's mechanisms for reversing or inactivating these phosphorylated proteins, the phosphatases. So remember, there's all these competing events to activate a system and inactivate a system. Don't get locked into this step 1, step 2, step 3, step 4. You must know these steps, but realize that they're part of a homeostatic system where you have activation, inactivation levels changing at all times. So if your activation level overwhelms the inactivation level, you have a higher concentration of the activated phosphorylated forms of these proteins, and the cell is going to produce a response. And these can cascade second messengers, third messengers, fourth messengers. Notice each time I skipped from the inactivated to the activated forms. Oh, excuse me. These are targets of this. There's an inactive form of protein kinase that becomes activated. But there's first chemical messenger system, second chemical messenger system, third chemical messenger, fourth chemical messenger, fifth, sixth. So it continues through many different levels of cascade. So leading ultimately to an appropriate cellular response. Now remember, in physiology we think of normal, so we want an appropriate cellular response. If there's a pathology or there's something wrong, a disease or disorder, maybe a genetic mutation in one of these the genes for one of these proteins, so that even though it gets phosphorylated, it can't become activated, then you do not have an appropriate cellular response. You have a pathological or imbalanced homeostatic system with the pathological or inappropriate response. The final example of the GPCR, the phospholipase C system, or maybe we'll look at the guanylyl cyclase also. The signal molecule again, so I'm just going to go through it as if it's the steps. Step 1, the signal molecule activates the receptor associated with the G protein. It changes as we described in the previous slide. The G protein activates phospholipase C, note the C, an amplifier enzyme. The amplifier enzyme, PLC, converts membrane phospholipids into diacylglycerol, which remains in the membrane, can move around, diffuse in the membrane, and therefore might have targets. And a cytoplasmic chemical signal, secondary chemical messenger, the inositol triphosphate, or the IP3, and it diffuses into the cytoplasm. So we now have the concentration of the extracellular signal molecule associated with the frequency of ligation, frequency of activation, and associated thereby by the intracellular concentration of the second messenger chemical. So you could get very tight correlations between the concentration of the signal extracellularly, the first signal, with the second signal intracellularly. Excuse me. So it might not be a one-to-one because this is a biological system. It might be amplified 1 to 30 or something, right? And then step 4, the diacylglycerol is mobile. Again, the fluid membrane model. It has targets within the membrane as well. An enzyme target is PKC, and it will phosphorylate proteins, and these phosphorylated proteins are associated with or directly responsible for the cellular response. Remember the cascading, so these phosphorylated proteins might have an immediate effect, or they might lead to a more enduring cellular response. The IP3 concentration in the cytosol is associated with the degree of ligation of a ligand-dependent calcium ion channel in the ER, endoplasmic reticulum membrane. Within the ER, you have calcium stores. It can't float around in the cytoplasm. It'll kill the cell. But it has very potent chemical signaling properties within the cytoplasm as long as it's free in the cytoplasm. So or cytosol, the fluid portion of the cytoplasm, right? So the cytosolic free concentration of calcium, or concentration of free calcium unbound, is very highly correlated with multiple cellular responses. So what they're not showing here is the reversal of this. Nearby there is an ATP-dependent calcium reuptake that is ongoing all the time. And there are other buffering mechanisms to remove the cytosolic free calcium because you can't let it build up. So these are very quick events. You get a burst in the IP3, a burst in the intracellular cytosolic free calcium, and then it's removed. It either binds to proteins to bring about the cellular response, or binds to proteins that buffer it, or is immediately taken right back up. So imagine a calcium comes out here, doesn't get very far, it's the first one to bind to the reuptake, it goes right back in. And because it occupied that one for that moment, here's a calcium that gets by it. Now we can talk about the receptor channel. So some second messengers create electrical signals. So here we see the extracellular signal molecules, either going to a G protein coupled receptor or to a ligand dependent ionotropic receptor or ion channel. So the metabotropic receptor can, through the G protein coupling, have an effect on the ion channel, or ligands can have an effect on it directly. So I think we're going to go through this in a series of slides, so let's do it that way. So here the extracellular signal molecules can, in this case, bind directly to the ion channel in the membrane and be associated with the state of open or close for this ion channel, and therefore be correlated with ion movement in and out of the cell. Ion movement, once this is open, is dependent upon electrochemical gradients. Chemical is the diffusion based upon concentration, and then the electrical is, we'll talk about more later, but that's where, you know, like, opposites attract, and the like charges, I've forgotten the word. You'll fill in the blank there, okay? Anyway, so if you got too many positives in here, it's just going to push each other out. Opposites repel or oppose each other. And if you got a lot of negatives out here, it's going to pull the positives out. So that's the electrical gradient part of it. So first, the receptor channels open or close in response to signal molecule binding. And then some channels are directly linked to G proteins. And the other ligand-gated channels respond to intracellular second chemical messengers. And collectively, we see a change in the membrane permeability to some type of ion species. Sodium, potassium, or chloride are the very common ones, but might be calcium. Creates an electrical signal across the membrane of the cell. Then you can have voltage-sensitive proteins that are associated with the cellular response. Continuing with this consideration of signal transduction, we see a summary map of the signal transduction systems. So we saw these signal molecules, in this case, going to the two types of receptors, the metabotropic or the ionotropic. This is a ligand-dependent ionotropic. There can be a ligand-independent ionotropic ion channel, like a voltage-sensitive sodium channel, or a voltage-sensitive or voltage-dependent potassium channel. Right? So there's no ligand that activates them, but some ligand-gated ion channels got to create an electrical potential event, so the voltage changes to favor either the open or closed state of the voltage-dependent ion channel. So back then, our ligand-dependent ion channel, or ionotropic channel, receptor, or metabotropic receptor that depends upon a ligand, and we have the G protein coupling that can act on enzymes, either activate or inhibit amplifier enzymes. Remember, we need both on and off switches. We need gas pedals and brakes to have a homeostatic system that reaches a set point and can increase or decrease and then be brought back to that set point. We activate tyrosine kinases, going to have similar regulatory events, gas pedals and brakes. We can alter cytoskeleton, as with the integrin receptors. We can get the second messengers activating third messengers, bringing about the phosphorylation of proteins, altering them so that we've got fourth messengers, and ultimately somewhere down the cascade there's going to be a cellular response with a change in motor proteins, like the contraction of a skeletal, smooth, or cardiac muscle cell, some type of enzyme activity, breakdown of glycogen in the liver cell to release glucose into the blood, membrane receptors and transporters, gene activity, and protein synthesis. So a wide range of events. And so here we can see ions in and out of the cell, a change in concentration that can create an electrical signal, triggering the release of calcium from the organelles. We saw a second messenger system that could do that as well, the IP3. The calcium can also alter proteins. The electrical signal can also alter proteins, bringing about cellular responses. So very complex, interrelated signaling systems, bringing about broad ranges of modulatory capabilities for cellular responses, many opportunities for regulation, also many opportunities for dysregulation, pathologies and diseases or disorders, or toxic effects, right? And also many possibilities for convergent of different signaling mechanisms. They're showing a signal molecule, but these could be two completely different chemical messengers. This could be a hormone, and this could be a neurotransmitter or something. So many opportunities for the interactions.